Hello everyone. In this lecture, I will briefly introduce the immunology. So from this chapter, we will learn about the immune system. We will introduce the main components of the immune system. The immunology is a branch of biology that covers the study of immune system in all organisms. The immunology measures and contextualize the physiological functions of the immune system in the states of both health and disease. Malfunctions of immune system in immunological disorders. Immunology studies the component and function of the immune system. The immune system has so many components and are typically cellular in nature and not associated with any aspect organ, but rather are embedded or circulating in various tissues located throughout the body. The molecules, the cells, tissues, and organs which provide non-specific and specific protection against microorganisms, microbial toxins, the tumor cells, the crucial to human survival. The major component of immune system is the lymphatic system, composed of the bone marrow, spleen, and the thymus gland, as well as lymph node and ducts. In addition, there are specialized blood cells called lymphocytes and leukocytes that working with the immune system. Their proliferation and differentiation in the central lymph node organ yield a variety of the T and B cell clones that seed into peripheral lymph node organs, whereas humoral and cell-mediated antigen-specific immune response occur. The thalamus is a specialized primary lymph node organ of the immune system. Within the thalamus, the T cells mature. T cells are critical to the adaptive immune system, while the body adapts specifically to foreign invaders. The lymph nodes are small glands that filter lymph, the clear fluid that circulates through the lymphatic system. They become swollen in response to infection and tumors. The lymphatic fluid circulates through the lymphatic system, which is made of channels throughout your body that are similar to the blood vessels. The spleen is the largest organ in the lymphatic system. It is an important organ for keeping body fluid balanced, but it is possible to live without it. The spleen is located under the ribcage and above the stomach in the left upper quarter of the abdomen. A spleen is soft and generally looks purple. Bone marrow is a sponge tissue inside some of the bones in the body, including the hip and thigh bones. Bone marrow contains immature cells called stem cells. The bone marrow is soft, gelatinous tissue that fills the medullary cavity, the center of the bones, and the two types of bone marrow are red bone marrow, known as myeloid tissues, and yellow bone marrow, or called fatty tissues. This slide is the introduction of the immune cells. There are several immune cells in the blood. The neutrophils are the most abundant type of granulocytes and the most abundant type of white blood cells in most mammals. They form an essential part of innate immune system, with their function varying in different animals. Eosinophils are a variety of white blood cells and one of the immune system components responsible for combating multicellular parasites and certain infections in vertebrates. In normal individuals, eosinophils make up about 1 to 3% of white blood cells. The basophils are the least common type of granulocytes, representing about 0.5 to 1% of circulating white blood cells. They are responsible for inflammatory reaction during immune response, as well as in the formation of acute and chronic allergic disease. The monocytes are a type of leukocytes, or white blood cells. As a part of vertebrate in the immune system, monocytes also influence the process of adaptive immunity. There are at least 3 subclasses of monocytes in human blood based on their phenotypical receptors. Macrophages and myeloid lineage dendritic cells are differentiated from the monocyte when monocytes infiltrate into the tissue. Mast cell is a migrant cells of connective tissue that contains many granules rich in histamine and heparin. Mast cells play a key role in the inflammatory process, especially in food allergy. T cell is a type of lymphocytes which develops in the thymus gland and play essential roles in the immune response. Groups of specific differentiated T cells have an important role in controlling and shaping the immune response. By providing a variety of immune-related functions, T cells are grouped into a series of subsets based on their function. CD4 positive and CD8 positive T cells are selected in the thymus, but undergo further differentiation in the peripheral to specialized cells, which have different functions. The B cells are a type of white blood cells of the lymphocyte subtype. They function in human immunity. Hematopoietic stem cells are the stem cells that give rise to other blood cells. This process is called hematopoiesis. This process occurs in the red bone marrow in the core of most bones. Hematopoietic stem cells give rise to different types of blood cells, in lines called myeloid and lymphoid. The myeloid and lymphoid lineages both are involved in dendritic cell formation. The myeloid cells include monocytes, macrophages, neutrophils, basophils, eosinophils, erythrocytes, and megakaryocytes, 2 planets. The lymphoid cells include T cells, B cells, natural killer cells, and innate lymphoid cells. A small number of hematopoietic stem cells can expand to generate a very large number of daughter cells. This phenomenon is used in bone marrow transplantation, when a small number of hematopoietic stem cells reconstituted the hematopoietic system. Since there are so many immune cells identified in immune systems, scientists develop a protocol called CD to distinguish them. The cluster of differentiation, which named as CD antigen, are defined when a surface molecule found on some members of standard panel of human cells. The cluster of differentiation is a protocol used for the identification and the investigation of the cell surface molecules, providing targets for immune phenotyping of cells. In terms of histology, CD antigens can act in numerous ways, often acting as a receptor or ligand, which is important to the immune cells. A signal cassette is usually initiate altering behavior of the cells. Some CD antigens do not play a role in cell signaling, but they do have other functions, such as cell adhesion, cell activation, and cell inhibition. This slide is the CD markers on white blood cells. Like the granulocytes, they are double CD45 and CD13 positive, and the monocytes is CD45 positive, CD14 positive. T lymphocytes is CD45 positive and CD3 positive. T helper lymphocytes is CD44 positive and CD3 positive and CD4 positive. The cytotic lymphocytes is CD45 positive, CD3 positive, and CD8 positive. The B lymphocytes is CD45 positive and CD19 positive. The natural killer cells is CD45 positive, CD16 positive, CD56 positive, and CD3 negative. There are two ways of immune response, including innate immunity and adaptive immunity. The innate immune system is essential, made up of barriers that aim to keep various bacteria, parasites, and other foreign particles out of your body, or limit their ability to spread and move throughout the body. The adaptive immune system, also referred as acquired immune system, is a subsystem of the immune system that is composed of specialized systemic cells and processes that eliminating pathogens by preventing their growth. We will discuss them in great detail in the next slides. The innate immune system is one of the two main immunity strategies found in vertebrates. The innate immune system is an older evolutionary defense strategy and is the dominant immune system response fungi, insects, and primate multicellular organisms. Innate immune system responds very fast and serves as first defense walls. Normally, the whole process takes within 12 hours to kill the pathogen. Adaptive immunity or acquired immunity is the protection mechanism from an infectious disease agent. As a consequence of clinical or subclinical infection with that agent, or by deliberate immunization against that agent which produced from them. This type of immunity is mediated by B or T cells following exposure to a specific antigen. It is characterized by specificity, immunoglobulin memory, and non-self recognition. The response involves clonal selection and the lymphocytes that respond to a specific antigen. The T cells and the B cells are the two major components of adaptive immunity. The humoral immunity or humoral immunity is the aspect of immunity that is mediated by macromolecules found in the extracellular fluid, such as secreted antibodies, complement proteins, and certain antimicrobial peptides. The humoral immunity is so named because it is involved substance found in the humus or body fluids. It contrasts with cell-mediated immunity. Its aspects involving antibodies are often called antibody-mediated immunity. The humoral immunity refers to antibodies production and the accessory process that accompany it, including Th2 activation and cytokine production, the germinal center formation, and the isotope switching, affinity maturation, and the memory cell generation. It is referred to the effective function of antibodies, which include pathogen and toxin neutralization, classic complementary activation, and uplink promotion of phagocytosis and pathogen elimination. The cell-mediated immunity is an immune response that does not involve antibodies. The cell-mediated immunity is the activation of phagocytes, antigen-specific cytotic T lymphocytes, and the release of various cytokines in response to antigen. Cell-mediated immunity, for which the protective function of immunization was associated with the cells. CD4 cells or helper cells provide protection against different pathogens. The naive T cells, which are mature T cells that have yet to encounter an antigen, are converted into activated effector T cells after encountering antigen-presenting cells. These APCs, such as macrophages, dendritic cells, and B cells, in some circumstances, loading antigen peptide onto the MHC complex in the cell. It turns presenting the peptide to receptors on T cells. The most important of these APC are highly specialized dendritic cells, conceivably operating solely to ingest and presenting antigens. Cells-mediated immune response lead a lot of cytokines. We will introduce the cytokines and how it works. Cytokines are a broad and loose category of small proteins, which is 5 to 20 kilo, which is important in cell signaling. Cytokines are peptides and cannot cross the lipid bilayer of the cells to enter the cytoplasm. The cytokines have been shown to be involved in the autocrine, paracrine, and endocrine signaling as immune modulation agents. Their definite distinction from hormones is still part of ongoing research. Cytokines include chemokines, interferons, interleukins, lympholucins, and tumor necrosis factors, but generally not hormones or growth factors. Cytokines are produced by a broad range of cells, including immune cells like macrophages, B lymphocytes, the T lymphocytes, and the mast cells, as well as endothelial cells, fibroblasts, and various stroma cells. The cytokines act through receptors and are especially important in the immune system. Cytokines modulate the balance between humoral and cell-based immune response. They regulate the maturation, growth, and responsiveness of particular cell population. Here is a summary of the function of our immune systems. Our immune system is essential for our survival. Without immune system, our bodies would be open to attack from bacteria, virus, parasites, and more and more. It is our immune system that keeps us healthy as we drift through a sea of pathogens. The major function of immune system is to protect the host from environmental agents such as microbes and chemicals, thereby preserving the integrity of our body. Thanks for your attention. Here is some reference.